Okay, perfect. طيب. Assalamu alaikum everyone. My name is Ibraheem Balkhi. I'm a third-year medical student, and today I'll be explaining two lectures. The first one is the regulation of gene expression, which is an important lecture, and I'll only explain the high-yield stuff and tell you where they usually ask and what questions they usually ask about. All right. Before we start, I would like to just give you a brief introduction for MOL, like a very quick one. So first of all, before starting MOL, as you know, you'll take it over 2 years. First year, over 2 semesters, sorry. So you need to start liking it. Even if you don't, you need to start forcing yourself to like it. And you need to start viewing it as an easy subject that you can manage. And I can't stress how important that is because you really want to avoid burnout, especially when you're in second semester. Then the second thing that I want to talk about is you need to start understanding MOL rather than memorizing it. And that is very important, and I'll show you when I explain the slides. And you need to know that each lecture is usually covers 2 questions. So when you study, you should keep that in mind. And I recommend that you stick to lectures and you watch YouTube videos if you don't understand something. For as in lipping cut, don't ever use it, in my opinion, because it might confuse you at first and it will take so much time that you don't have. Okay. So also regarding medicine, I want you to know that studying isn't all. It's extremely important that you understand medicine and not memorize it. And this might seem funny, but it's not. It's actually the way to go about medicine, in my opinion. And some student might fail through the first year, and I want you to know that that's not the end. A lot of students have failed and managed to pass and graduate. And yeah, so let's start. So before I start, I want to introduce something. Oh, can you guys hear me clearly and see my screen? Let's confirm because I don't want to. Okay, perfect.
Where the fuck did Charlie go? Yo, where did Charlie go?
Okay, we apologize for that. Okay, so before we start, I want to introduce something, and it's called the big picture approach. And in my opinion, before starting anything or any concept in medicine, you need to see it as a whole. You need to see what they're talking about. Why is this there? Where is that? Get me? So in order to further understand the big picture approach, I will apply it to this lecture. So today we're talking about the regulation of gene expression, or how are genes regulated. And to start this, we will talk about, okay, that's right, fine. Okay, so, and to start this, we will talk about, I'll show you what the whole lecture will cover. So first of all, DNA is regulated on many levels, starting with transcription control. So transcriptional control, some genes might be transcribed while others aren't. And this happens by 2 mechanisms. This can be chemical control by either demethylating the DNA, hence expressing it, or acetylating the histones. That's a very important thing to distinguish. You demethylate the DNA and acetylate the histones, and this is used to express the gene, and hence to transcribe it. And then we'll talk about transcription factors and enhancers. So by these 2 ways, you regulate how many mRNAs you produce, how many protein you produce, and hence the phenotype. And that's the main goal of this lecture. Then you had, let's say you transcribed it, everything is good. You're talking about the primary RNA transcript. Now, what's the difference between primary mRNA transcript and mRNA? It's the fact that mRNA, or mature mRNA, is after splicing. So you know that DNA is made of exons and introns, and then when it's transcribed, it forms exons and introns. Now you want to remove the introns. That's the first thing that happens in splicing. The second thing is you want to rearrange the exons, okay? And this produces a mature mRNA. Now this is a form of regulating gene expression, because if you rearrange the exons in different ways, you get different mRNAs. So that's another way to rearrange gene expression. Now, RNA transport into the cytoplasm. Can you make sure everyone's muted, please? Okay. So RNA transport into the cytoplasm. not all the mRNAs are transported into the cytoplasm, and hence not all of them are translated. That's another way of controlling it. Stability control. The mRNA has a certain lifetime. So if it's in the cytoplasm for long, it gets transcribed more, and hence more proteins, more phenotype. Okay, that's another form. Translational control. Sometimes proteins bind to the mRNA, inhibiting its translation. And lastly, not every protein that is made, okay, is used. That's another form of control. Now, this whole thing is an example of epigenetic inheritance. Okay, this is the entire lecture. Okay, I'm gonna go over it one by one, make sure you understand everything. But if you understood this, you're good to go. Okay, so epigenetic inheritance. Now, usually when we talk about inheritance, we talk about how genes are inherited from parents to children. But when we say epigenetic inheritance, we're talking about the mechanism that alters the phenotype. For example, let's say you had a DNA that says CGAT. Okay, let's say this is a gene that forms a certain protein. Now, just because you have this gene doesn't mean you form that protein, and hence doesn't mean that you form that phenotype. Okay, they thought previously, oh, I have this sequence, so I'm going to form this phenotype, but that's not the case. What if this DNA, as we're going to learn, is methylated? Methylated means the whole gene is inactive, so you don't get the protein. Okay, so just because مثلا your DNA says you have CGBAT TAACG, for instance, doesn't mean that it will produce all the phenotypes. It all depends on the mechanisms that regulate that sequence. That's epigenetic inheritance, and it's a very important point that they ask about in the exam. Now, I want someone who is watching this lecture to start recalling the questions that they're going to come. Okay, so the first one that they usually ask about is the definition of epigenetic inheritance. I'll make sure I explain it clearly when the slide comes to it. So that's a very important thing that they ask about. Okay, so someone please keep track of everything. So my goal from this session is to tell you what questions are probably coming, the high yield concepts, make sure you understand them, and give you maybe a way to study. So on this slide, you always skip it. And this slide, you always skip it. So when we talk about gene expression, as you know, all cells have the exact amount of genes, okay? But why is a liver cell different from a pancreatic cell? It's because the liver cell has some genes that are turned on that are different from the pancreatic cell. And this brings us to the first concept that you need to understand, and it's called cell specialization. And this is a high yield concept that you need to understand. So all cells have the same exact amount of genes, but the only thing that's different is the rate of transcription of those genes and the genes that are on and the mRNAs that are expressed and translated. So that's important to understand. So all cells are genetically identical, but different gene expression, okay? Again, this is the big picture. Now, as you know, you look the way you look because of the proteins that you have, okay? And where they're distributed, the amount. I'll give you a small example, and you can obviously scale that according to the billions of genes that you have. So for instance, melanin. The more melanin you have, which is a protein, the darker your skin color is, okay? So what if the gene of melanin is methylated? It won't be transcribed, hence you look white, okay? And what if the gene of melanin is there, but in one person it's enhanced or more protein is made, and in the other it's not? So that will create different skin tones. So that's a small example, and you can obviously scale that as you wish. All right. So the first thing we're going to talk about, the first example of epigenetic inheritance is transcriptional control, which means controlling the phenotype on a transcriptional level. And the way you control phenotype on a transcriptional level is by two ways. First of all, you either acetylate the histone, so you acetylate the histone, demethylate the DNA, okay? I don't want you to say that you acetylate the DNA and methylate the histone, okay? It's completely different, okay? So you acetylate the histone, demethylate the DNA, and this will cause the expression of that gene, okay? And the reason this happens is because this causes the DNA, I don't know how to draw DNA, okay, to unwind. So when it's winded up, okay, you don't get transcription. But when you acetylate the histones, it unwinds, as in, okay, so here. So usually DNA is winded here, but when you acetylate the histones, you get unwinded DNA, okay? So now RNA polymerase can come and transcribe this part. So that's the first thing. Now let's say you had a DNA sequence, and this
Assalamu
DNA
alaikum.
sequence is methylated. Alaikum salam. Type. And this DNA sequence is methylated. If this DNA sequence is methylated, RNA polymerase cannot bind. But if you demethylate it and it becomes like this, RNA polymerase can bind. So that's a very important distinction that you should come to conclusion to. And it's the basics of this lecture. So that's one. We have done the first part of transcriptional control. The second thing is the DNA binding proteins. Just because you have a DNA that is not methylated and is exposed to RNA doesn't mean that it will get transcribed. In order to transcribe a certain gene, so you have a gene, all right, and you have before it a promoter sequence, okay? This promoter sequence needs to be activated in order to transcribe the gene. So if this promoter sequence wasn't activated, you want this gene, you control it at a transcriptional level. We'll talk more about this, okay? I don't want to explain this here, I'll explain it later. But let's talk about a quick structure for the DNA. This is very important. It actually came in our exam. So this is the second point, second question that they can ask. What's the structure of a nucleosome? So let's break it down. You have histones. So you have 1, 2, 3, 4, 5, 6, 7, 8 histones. Let's talk about the ones the four on the top. So they're made of H2A, H2B, H3, and H4, and they're repeated. So that makes an octamer. So an octamer is 8 histones. This octamer gets wrapped around by DNA, it's called a nucleosome. Now that you're oriented with the structure, let's make it even more complicated. Let's say each nucleosome is connected by something called linker DNA. And in the linker DNA, you can find H1 histones. Now, when DNA wraps around H1 histones, you get less transcription. I don't want you to get confused, but I want you to get oriented. So when I tell you what an octamer is, you tell me it's H2A, H2B, H3, and H4. These are all proteins, okay? And there are 2 of each, okay? If I tell you H1, you tell me it's found in the linker DNA between nucleosomes. So that's a very important orientation. And I'm not telling you to memorize this, I'm just telling you to be oriented. So understand this. But we've talked about DNA methylation and histone acetylation. So what do we want? What do we actually want? We want to transcribe. So when we want to transcribe, we want to expose as much DNA as possible. And the way you do this, you acetylate. Acetylate means you add an acetyl group, which is, I think, is a C double bond O H. If I'm not, that's not the point. But the point is you add a group to the histones. When you do this, you separate the histones or the nucleosomes from each other, exposing what the linker DNA, which can now be transcribed into genes and getting the proteins that you want. Now let's get into more details. Where do you acetylate those histones? You acetylate them at something called the charged lysines. Lysine is an amino acid, and it's found in the histone tails. So how can they ask about this? They will tell you, oh, if I want to make more of this gene, they're not going to give you a whole scenario, but you need to understand from that scenario that they want to turn on a gene. So they'll tell you, do you deacetylate the DNA? Do you acetylate the DNA? Do you deacetylate the histone, acetylate the histone? Is it at the lysine? Is it at the proline? Is it at the tail, or is it at the head? So these are all keywords that they can play around. So here's what you do. If you want to turn on a gene, you acetylate the lysine in the tail, okay? Now you have to memorize this, okay? But you need to understand that when you do this, the positive charges will repel each other, exposing the DNA, and you get transcription. All right. Next up, when you methylate the DNA, now the DNA should be like alone, stranded. When you methylate it, you add a methyl group. RNA cannot do it. So I don't want you to memorize, okay, DNA methylation, demethylation, which one turns on, which one turns off. Because if you do this 100% in the exam, you're going to get lost. Do we acetylate the DNA or the histone? Do we methylate the DNA or the histone? So focus. So acetylate histone or demethylate the DNA. And this makes sense because if the DNA has something added to it, such as a methyl group, it won't bind and you won't turn on the gene. And this whole thing is an example of epigenetic inheritance at the transcription control. So do we get the big picture? That's a very important thing for us to proceed. Like, I've been talking a lot about epigenetic inheritance. The reason is because they really love asking about it. I think they asked us about it in the midterm and the final, both. So when I talk about epigenetic inheritance, it's the phenotype. Now, you know what phenotype is. Phenotype is the way, this is the characteristic. So eye color, skin color, shape, everything, okay? It's the phenotype that is caused by not just the genes, but the mechanisms affecting the genes, okay? So previously they considered inheritance, and if you had a sequence, you would get a phenotype. But now they know more. They know that not just because you have a sequence, you'll get the phenotype. If that sequence is regulated, if that sequence is turned off, you won't get the protein, you won't get the phenotype. If that sequence is turned on, it's different from if it was turned on with an accelerated rate of production of protein. Get me? So these are all ways in which a different phenotype is produced, okay? So this is epigenetic inheritance. It's the inheritance due to mechanisms and not due to sequences, okay? So they'll play around a lot. So they'll give you a scenario and then they'll tell you, no, the question's point is what's the definition of epigenetic inheritance? And they'll change a lot in the words, but you need to understand in which choice relates it to mechanisms and not to sequence. So it's the one inherited, it's the phenotype by the mechanisms and not nucleotide sequence. Good. So that's a question that's coming. So I've talked about two questions. The first one is the definition of height. And the second one is this one, they'll play around with acetylation. So what do you acetylate, when do you acetylate? And the third one is actually the structure of a nucleosome and an octamer and an H1 and the linker DNA. So these are three questions. So from this lecture, two questions are coming, okay? But these are possible questions that they love to ask about. Okay, so we've talked about the regulation of transcription. Let's talk more about it. So we've talked about the chemical changes. Now let's talk about transcription factors. So just because you have a sequence doesn't mean that it will get transcribed. All right. So there are two types of transcription factors. You have the general transcription factors and you have the specific transcription factors. Now, this is a weak point of a lot of medical students. So they love to ask about this. Add this to the list. So how do they ask about this? They ask, what's the function of transcription factors? Okay. So basically it depends. So you have general transcription factors. You know what? I'll talk about this later because there's a slide that shows better pictures. But let's talk about this now. Okay, let's leave this for now. I'll talk about this. So here, I don't know why they mentioned this because it's out of place. Okay, but I'll explain it. So basically, the difference between prokaryotic cells and eukaryotic cells is that prokaryotic cells have different translation binding sites. So this is an mRNA, and this is a eukaryotic mRNA. So for one mRNA in a eukaryotic cell, you can produce one protein, okay? Because there is only one binding site for the ribosome. However, in prokaryotic cells, one mRNA can produce different proteins, okay? So one mRNA produces protein 1, protein 2, and protein 3. This is called polycistronic. Memorize it. This is very important, but it's not, I think they mentioned it in a different lecture. And they asked about it in that lecture. And this is called, and this is called monocistronic. Monocistronic, that means the mRNA codes for one protein, and it's found in eukaryotic cell. If you know that, you can leave it. What's an operon? An operon is, you can think of it as a gene, okay? So it's a functioning unit of DNA. So that explains that, and I'm sorry, and you had to endure that because that's out of place in the lecture, in my opinion. Okay, one thing that I actually want to make clear is that they usually ask about if they give you a DNA sequence, so C, A, T, and G, okay? this is 5 prime, and this is 3 prime. And they tell you transcribe it. So what you usually do is you do, oh, C, that's a G, and then A, that's a, damn, I forgot. So A, that's a U, and T, that's an A, and G, that's a C, okay? And you put this as an answer. One thing that they like to trick you about, that's not the answer. So this is, when you transcribe it, the mRNA flips actually. So this is the 3 prime end and the 5 prime end. And it's known in biochemistry that whenever you write a nucleotide sequence, you always write it from 5 prime to 3 prime, okay? So you're supposed to write it C, A, U, G. Okay, that's a question that's 100% coming, but it's not in this lecture. I just thought I'd mention it for, yeah. Like, okay, now let's talk about transcription factors. Before we start, we have a gene, okay? The gene codes for a protein. Within this gene, you have different exons and the black line here, which represents introns. And these will be removed during RNA splicing, and the exons will be rearranged during RNA splicing. Before this gene, you have a promoter region. Now, this promoter region needs to be activated in order for transcription to happen. That is, if the DNA isn't methylated or if the histones aren't deacetylated. So if all the conditions are right, the promoter region needs to be activated. And the way it's activated is by transcription factors binding to the promoter region. Now, these transcription factors are called general transcription factors, which are the first transcription factors which bind to the promoter region, allowing for the transcription of the gene, okay? And then you have specific transcription factors which can bind to the general transcription factors and then bind to either silencers or enhancers. If they bind to silencers, they're gonna decrease the rate of transcription. If they bind to enhancers, they're gonna increase the rate of transcription. Add this to the list. They usually ask about what's the function of enhancers. And you're gonna choose the one that says it increases the rate of production of mRNA, which should make sense. So enhancers bind to the transcription factors, which then bind to the RNA polymerase, increasing its rate, okay? So that's a very important distinction that you should have. And that's the only important thing that you should know from this slide. طبعا here, just for reference, I'm gonna quickly go over it, except it's not important. So they talk about cis-acting elements. The cis-acting elements are the silencers, enhancers, promoters, because they are on the DNA. And then the trans-acting elements, they are the transcription factors, which are proteins. They're called trans because they're not part of the DNA. It's not important. I don't want you to focus on this. But what I want you to focus on is the fact that enhancers increase the rate of mRNA production by binding to transcription factors, which will then increase the rate of transcription. I'm sorry if this is taking too long, but I'm explaining the whole lecture in the first part. When we reach the end, we will skip some slides because we've already explained them. Okay, so this is a gene. We've said it's made of exons, introns, and Now, an enhancer is usually located upstream. This is a very important distinction. An enhancer is located upstream, and it's usually 100 to 200 pairs long, and it's located usually 50 kilobases away from the promoter region or the gene. All right, so you have this is the DNA, this is the gene, 50 kilobases. You have an enhancer. An enhancer is a DNA code, by the way, if you don't understand, if you didn't know. So the enhancer binds to transcription factors, a lot of transcription factors, which are specific transcription factors, which this entire complex of enhancers and transcription factors stimulates the RNA polymerase, increasing its rate. Okay, silencers decrease the rate, and it's usually found command upstream. Promoter regions are usually located very close to the gene, so 200 bases upstream. When we say upstream, yani within 200 bases. Like if this is 3 prime, this is 5 prime. So upstream is towards 5 prime. Okay. Like, and this is what we've talked about. We've talked about general transcription factors, which are needed to initiate, which bind to the promoter region and initiate transcription. Specific, they control. They either increase the rate or decrease the rate. Okay. and they bind to enhancers or repressors. And that enhancers are distal control elements, and proximal control elements are the promoters. Okay, proximal يعني قريب, distal is far. Okay, so we've talked about this. Okay, so we have, let's say this is an enhancer. This part of DNA is an enhancer. So it's very important that you understand enhancers are a DNA sequence. Okay, this is a DNA sequence. The transcription factor has a DNA binding site and a RNA binding site, or the activation domain as they call it. So the activation domain binds to RNA polymerase, okay, which stimulates transcription or initiates it, depends on whether this is an enhancer or a promoter. So I just want you to know that this is the structure and it makes sense. And that the DNA binding site is made of zinc fingers. That's all you need to know. The DNA binding domain is made of zinc fingers. Why? I don't know. It's not important. Type. This is an example, a real life example of how enhancers are important. Now, let's quickly overview it. So let's talk about cortisol. Now, cortisol is a lipid soluble chemical. So it can enter, so it's hydrophobic. So it can enter through the phospholipid membrane. When it enters, it binds to this enhancer called GRE. And as we said, enhancers increase the rate of transcription. So this entire complex of cortisol and GRE is now, so cortisol here acts as a transcription factor. It's a specific transcription factor. It will go ahead and increase the rate of the translation of transcription of. Now, I don't know if you took this, but PepsiK is an important enzyme in the formation of glucose or gluconeogenesis, okay? So you want to increase glucose. You usually want to do this when you have hypoglycemia, when you have low glucose in the blood. When you have low glucose in the blood, you want to produce more glucose. So your body stimulates the release of cortisol, or a stress hormone, which then binds to the enhancer and increases the rate of PepsiK. PepsiK is the rate limiting enzyme, and it produces more glucose. That's one mechanism, okay? Another mechanism, you know, glucagon. Glucagon is a hydrophilic protein. Since it's hydrophilic, it cannot enter through the basement membrane. However, it can bind to receptors. Now, this creates an intracellular change that I 100% don't want you to know, or they won't ask about, because this is not the point of the lecture. Okay, but what the end result is the production of CREB. CREB will bind to another enhancer called CRE, and this entire complex will go ahead and increase the rate of production of mRNA, which will then be translated into PepsiK, forming more glucose. So, and you should know this. So when glucagon is released, it usually stimulates the liver or hepatocytes to secrete more glucose. Okay, so Ibrahim, this is so confusing. I don't understand anything. Here's what I want you to know. I want you to know that to understand this as a whole, okay, just have it in your mind that, you know, transcription factors are usually not necessarily in the nucleus. They can come from outside, or they can be stimulated from outside, okay? And you understand the mechanism of how transcription factors work. They bind to enhancers and they increase the rate of, I'm stressing on the word increasing the rate because they ask about it a lot. Okay, so this explains all of this, and explains all of this, and explains all of this. Okay, what we just said. You don't have to 100, like, you definitely don't need to know this. You do not need that increased CMV, protein kinase A, or CREB, because you will know it later in your second year or third year. Type. Okay, so this whole thing was just us controlling transcription. Okay, and I want you to understand that. And it's the first part of epigenetic inheritance. Now, let's say we've gone through all this hassle. We've produced the mRNA. Now, we want to fine-tune the gene expression. Okay, and this includes, we've produced the mRNA. And this is called primary mRNA. And it has the introns, which you can see here. And yeah, it has the exons, which you can see, they're numbered here, okay? Now, just because we produce the mRNA doesn't mean we can't affect the phenotype. We can. And this is done, like, one of the first things is RNA processing. RNA processing, as you know, it's called alternative RNA splicing. You remove the introns and you rearrange the exons. Okay, and that's one way of controlling the phenotype. That's an example of post-transcriptional modification. So after transcription. I hope I'm making sense and I'm not going too fast. Okay, the other way is just because you have spliced the RNA and produced and made it doesn't mean that it will form the protein, because the mRNA can be immediately degraded in the cytoplasm. Okay, so the lifespan of an mRNA determines if it's going to produce a protein or not. So if an mRNA has a high lifespan, it will stay in the cytoplasm for a long time. It will produce more proteins. And if it doesn't, if it gets degraded immediately, it won't produce the proteins. And they say, the half-life is determined by the UTR at the 3 ends. So you have the mRNA, mature mRNA with the 5' end and the 3' end. They say that there is an untranslated region here. Okay, so this region, so as you know, because in the mRNA, you have the part which will form the protein, and you have a UTR here and a UTR here. A UTR is an untranslated region. That means it won't get translated. So, so for example, in the 5' UTR, there will be sites for the ribosome to go and bind and start the translation of this part. And it will stop here. There will be a sequence that will stop it here. Okay, so another thing to add, and this is actually very important, is that the UTR in the 3' end will determine the half-life. So if it had a sequence which promotes degradation, it will produce less protein. So that's one thing that you should keep in mind. Okay, just because you have an mRNA doesn't mean it will get translated. This mRNA, as we said, we have a 5' UTR, and this 5' UTR has a region for the ribosome to bind. This region might be blocked by a protein. Okay, this region might be blocked by a protein, so it won't get translated. Okay, so that's an example of an initiation of translation. Okay, and one of the most important examples that you should 100% know is ferritin. Now, ferritin, which is something that we studied a lot in heme, and it's very important. It's a protein that binds iron. So when you have a lot of iron, you don't want that in the cell, because it will deposit in the mitochondria, it will deposit everywhere and cause a lot of problems. So you want to control that. So ferritin is a protein that binds to iron and regulates this whole thing. Now, when usually the ferritin mRNA gene is found in the cytoplasm, but however, usually it's bound to IRE binding protein. IRE binding protein, so you have this part, which is called an iron response element, and it's a sequence of mRNA. Okay, call it, for instance, X. Let's call this part X, for instance. Okay, now you have an X binding protein. Does that make sense? Now, when X binding protein is bound to X, you don't get the translation of the mRNA. However, when there is increased intracellular iron, what happens is that iron will bind to the X binding protein, or the IRE binding protein, removing it from the IRE. And now the ribosome can bind and translate it into the ferritin protein, which will then bind the iron as a mechanism of regulating the concentration of iron in the cell. Does that make sense or not? Because I feel like I lost you guys. Does that make sense? Okay, I'll repeat it again. So we're talking about the expression of a phenotype, or the expression of a protein. One of the mechanisms in which you can regulate the protein production is by controlling whether this mRNA gets translated or not. And this is done by some proteins that bind to the mRNA, deactivating it. For example, one of the proteins is called IRE binding protein. It will bind to the mRNA, okay, inhibiting its translation. So when you have IRE binding protein, this mRNA will not get translated, okay? And once you have increased intracellular iron, the intracellular iron binds to the IRE binding protein, removing it from the mRNA. Now you have the mRNA alone. And just now, since it's free, it can get translated by the ribosome. Once it's translated, this is a ferritin gene, it will produce ferritin. Okay, now ferritin binds to iron, and this is a mechanism of protecting the cell against increased iron. And this is just an example for you to understand and how the mRNA is controlled by proteins which control its translation. Okay. At the end, I will make sure I repeat everything so that you understand it in 5 minutes. Okay. Another mechanism in which we can regulate the translation is by something called RNA interference. Now, RNA interference is something completely different from mRNA or tRNA or rRNA. They're something different, okay? They include microRNA and small interfering RNA. I'm going to make this as simple as possible. So you have microRNA and small interfering RNA. Now, these are different types of RNA which can bind mRNA, inhibiting it. Does that make sense? If you understand this, that's more than enough. If you understand that small, what's it called, small interfering RNA or siRNA and miRNA are more than enough. And this is the way they work. You 100% don't need to know this, okay? So the way I'm gonna go over it extremely quick. So just because you don't have to know this doesn't mean that you can't keep it in the back of your head. So I'm going to go over it quickly. So miRNA binds to Argonaute protein, and this entire complex binds to the mRNA, degrading it. So it gets degraded. So you don't have the mRNA, you don't have the protein. And that brings us to the end of the lecture. Okay, I'm going to quickly go over the slides to make sure you know the point which I explained. Like, actually, I want to go over this slide. That's it. I just want to go over this slide. Like, so we've talked about transcriptional control. We've mentioned DNA demethylation and histone acetylation to boost gene transcription. We've talked about transcription factors. We've talked about enhancers, their role in increasing the rate of transcription, and how some general transcription factors are needed to initiate transcription in the first place by binding to the promoter region. You know, understand that enhancers are part of the DNA, which are located upstream, and they regulate the rate of production. We've talked about RNA processing and how the removal of the introns and the rearrangement of the exons can produce different proteins and different phenotypes. We've talked about how the RNA in the cytoplasm has different things that prevent its translation or promote its degradation. We've talked about translational control. In the next lecture, we'll talk about post-translational control and how proteins are actually transported throughout the cell. This whole thing is epigenetic inheritance. Now, before I go to questions, I just want to give you a quick example explaining the importance of epigenetic inheritance. It was noted that in one of the European countries back during the war, that one of the countries their population was starved to death. Okay, as a consequence, the second generation, they found that the genes that the people from the second generation have a higher tendency to keep food or to have a less metabolic rate. So they want to keep the food in their bodies, okay, or have a lower metabolic rate, okay, so that they don't use food. So what am I talking about? I'm talking about how something that is not related to the DNA sequence, because of the stress on the population of the first generation, this affected the inheritance in the second generation, and it's not related on the sequence. So that is epigenetic inheritance. You can look into it more if you want, but that's an example. You don't have to know this. I just wanted to make it more clear or more confusing. So questions for everyone else. We take a 5-minute break and we answer some of the questions, and then we come back in 5 minutes. So 6:45, we'll start the second lecture.